everyone, welcome to today's webinar on behalf of Biocytogen. We are thrilled to have you join us as we explore exciting preclinical mouse models, advancing small nucleic acid therapeutics and gene therapy. Today, we are pleased to introduce one of our experts who will be sharing insights that can support and enhance your ongoing research program. Before we begin, allow me to briefly introduce myself and our speaker. My name is Xiaoxuan, and I'm part of the marketing team here at Biocytogen Beijing. It is my great honor to introduce today's speaker, Dr. Jinyi Ma. Dr. Jinyi received her Ph.D. in Tianjin Medical University, majoring in mechanistic study of pyroptosis and the development of single-domain antibody screening. She subsequently joined Biocytogen as a product manager in the BioMice Product Department. Dr. Jinyi manages the development of humanized mouse models for metabolic diseases and small nucleic acid therapeutics. A series of humanized mouse models has been successfully implemented across diverse therapeutic areas, including cardiovascular diseases, obesity, diabetes, and non-alcoholic steatohepatitis, NASH. So now I would like to invite Dr. Jinyi to begin her presentation. Participants may ask their questions in the Q&A box or drop us an email, and our representative will address the questions by email respectively. Thank you, and I hope you enjoy the presentation. Let's welcome Dr. Jinyi.
My name is Jinyi Ma. I'm the product manager from Biocytogen, and today I will introduce our humanized mouse models for small nucleic acid drug and in vivo gene therapy. Our Biocytogen consists of two segments, Ren Biologics and BioMice. Ren Biologics is a scaled antibody discovery platform, and our BioMice can provide gene editing services and gene editing mouse models for basic or preclinical research, as well as we will provide our pharmacology services. And our company is based in Beijing, and we also have the branch offices in Jiangsu, Haimen, Shanghai, Boston, Heidelberg, and San Francisco. Here is my agenda today. Today, we will explore our humanized mouse models or gene mutation models and how they can be used in small nucleic acid drug and in vivo gene therapy. First of all, I will briefly introduce the mechanism of small nucleic acid drugs. We know small nucleic acid drugs is composed of nucleotide, represent a novel class of pharmaceuticals that differs from the conventional small molecule drugs and antibody drugs. And they can work by targeting the specific gene sequence to modulate gene expression and protein translation. And small nucleic acid drugs include antisense, ASO drugs, and small interfering siRNA drugs, and microRNAs, and aptamers. And two major types of FDA-approved research are ASO and siRNA. And we can see siRNA is a class of double-strand RNA molecule, and it can form the RNA-induced silencing complex and leading the RNA degradation. And ASO is a class of single-strand sequence, and it can bind with the pre-mRNA or mRNA, and leading to the RNA degradation, translate inhibition, or exon skipping. And because of the specific ability of small nucleic acid drugs, they have the potential to develop the pharmaceutical drugs for undruggable disease, such as rare disease and cardiovascular disease and some chronic disease. But it also has some side effects or have some challenges, such as the low drug delivery efficiency except for the liver, or the toxicity because of the off-target or the immune response or the low tissue and cell penetration. So for the preclinical research of small nucleic acid drug, the model choice is really important. And here is the process of how we develop a humanized mouse models. And in the first step is gene targeting. because small nucleic acid drugs can target exons, introns, or UTR. So most strategy of our humanized mouse models are in situ genomic replacements. And after we obtain these mouse models, we will conduct the protein and mRNA expression data. And we can compare the expression level between wild-type mice and our humanized mice by ELISA and qPCR. And we will compare the expression stable in between different ages. And in the in vivo efficiency test, we will conduct mRNA or protein knockdown test by using a positive drugs from the pattern. And in the last, we will conduct a functional analysis and in vivo efficacy by establishing a disease model. Here is the product list of our humanized models for small nucleic acid drugs across different disease areas, such as eye disease and chronic disease, such as hyperlipidemia and obesity, as well as neurological disease, and for lung disease or liver disease. And now we will explore the data and I will show you the case study for small nucleic acid drugs. The first disease area is cardiovascular and hyperlipidemia. The first popular target is PCSK9. We know PCSK9 is expressed in the liver, and it can target the specific receptors such as LDL receptors, leading the LDL receptor degradation in lysosome. And after we inhibiting the PCSK9, it can induce the LDL receptor recycle to the cell surface and leading the clearance of LDL particle. And inclisiran is the first-in-class siRNA drug targeting PCSK9, and it can induce the clearance of LDL cholesterol. Based in this mechanism, we develop our humanized PCSK9 Mice Plus. Here is the gene targeting strategy. In this mice, the genome of mouse PCSK9 include the 5' and 3' UTR was replaced by human genomic, including the 5' and 3' UTR in our humanized PCSK9 Mice Plus. And here is the model validation data. And here is the RT-PCR result for the mRNA. And we can see here is only human PCSK9 in our humanized PCSK9 mice. And here is the Western protein expression results by ELISA. And we can see here is only mouse PCSK9 in wild-type mice, and here is only human PCSK9 mice in humanized mice. And here is the in vivo inhibitory efficiency test. And in this study, our humanized PCSK9 mice plus were randomly divided into two groups. And in group 1, these mice were treated with PBS as control. And in the group 2, these mice were treated with siRNA drug targeting PCSK9. And we collect the blood of these mice at different days. And we can see in the figure B, the protein expression level of PCSK9 was significantly reduced in different days, which means our humanized PCSK9 Mice Plus can provide a powerful platform for the preclinical research for small nucleic acid drugs. Another target is LPA. We know lipoprotein A is an independent cardiovascular risk factors determined by KIV2 copy number. And lipoprotein A is formed by ApoA and ApoB. And for the complex of lipoprotein A in the first step, the lysine residue at N-terminal of ApoB are non-covalently bind to the lysine binding site located at the KIV7 and KIV8 domain of ApoA. And in the second step, A covalent disulfide bridge is established between the KLV9 of ApoA and ApoB. Because we know LPA gene is exclusively expressed in the primate, so our humanized LPA mice is targeting by the full coding sequence of human LPA with one copy number of KLV2, which is driven by ALB promoter, was inserted into the ROSA26 leukocyte. And here is the protein expression data of human ApoA in our humanized LPA mice between male and female across different ages. And we can see the protein expression level is stable, and we can see the protein expression level in male is higher than female. So we can try male to conduct the inhibitory test. And in the right, our humanized LPA mice were treated with an siRNA drug, olpasiran, which is an siRNA can target mRNA of LPA and leading to the decrease of concentration of lipoprotein A. And we can see in the figure A and the figure B, the concentration of ApoA was significantly reduced at different days after siRNA drug treatment. And according to the report, the PCSK9 inhibition can increase the clearance of lipoprotein A by increasing the expression of LDLR and VLDLR. So we cross our humanized LPA mice and the PCSK9 mice for the combination treatment or the double target drug treatment. And in this double humanized mice, we treated our humanized mice with siRNA drug targeting PCSK9 and LPA. And we can see the concentration of PCSK9 and LPA was significantly reduced after treatment. Apart from small nucleic acid drugs, we know there are still some small molecule drugs are still under development, such as mevalopin, which is a first agent developed to lower the lipoprotein A by targeting the assembly of ApoA and ApoB. So we also cross our humanized LPA and ApoB mice for more drug forms, such as small molecule drugs. And we can see in our double humanized LPA and ApoB mice, we can detect LPA complex in the serum, and we couldn't detect the lipoprotein A complex in the single target humanized mice, which means the lipoprotein A can successfully formed in our double humanized LPA and ApoB mice. And here is the product list for more cardiovascular disease. Except for the PCSK9, we also have the targets such as APOC3 and hypertension target AGT, REN, and some target for thrombosis such as F11 and F12. Another disease area is obesity. As we know, GLP-1 treatment is one of the most important targets for the pharmaceutical market, but it also has some side effects, such as the suboptimal GI tolerability and the lack of durability of weight loss and the issue of muscle preservation. So we still wanted to explore more target for obesity treatment. This slide shows the obesity drug pipeline, and we can see IHBE is an important target for some small molecule drug companies. Here is the mechanism of IHBE. We know IHBE encodes a protein called actin A, and it's excreted from the liver, and it can target receptor ALK7 at adipose and leading to the block of fat burning. So the silencing of NHBE restart and maintain the lipolysis. Another reason to develop NHBE as a target because of the individual who have a protective loss of function mutation in NHBE will have a healthier cardiovascular cardiometabolic profile. Here is the gene targeting strategy for our humanized NHBE mice. The exons and introns of the mouse NHBE gene encode the full-length protein, and the 3' UTR were replaced by human NHBE gene, including the 3' UTR. And here is the MRI result by RT-PCR, and we can see here was only mouse NHBE in wild-type mice, and here was only human NHBE mRNA, our humanized mice. And the figure B shows the protein expression in the serum in male and female NHBE mice. And antibody is cross-reactive with both with human and mice. And after we have the humanized NHBE mice, we conduct mRNA knockdown test by treated with an siRNA drug target NHBE. And after treated these drugs, we collect the liver tissue at day 14, and we compare the mRNA expression level between the control group and siRNA drug treatment group, and we can see a significant decrease of the mRNA of NHBE. After that, we conduct our in vivo efficiency data in our humanized NHBE mice. And our humanized mice were treated with high-fat diet to induce a DIO model and treated with the siRNA drug target NHBE. And we can see in the figure B, the body weight growth can significantly be inhibited by the siRNA drug compared with the high-fat diet ceiling drug. And we also have MRA results. We can see after siRNA drug treatment, the MRA level of human HBE was significantly reduced, as well as the liver TC and liver TG also can be reduced after the siRNA drug treatment. Here is the 3D reconstruction analysis of lean tissue and adipose tissue by micro-CT after treatment. And we can see the lean tissue weight and the ratio between lean tissue and the body weight will not decrease after siRNA drug treatment, but the adipose tissue weight was significantly reduced, which means the siRNA drug target NHBE will not decrease the lean tissue, but can decrease the adipose tissue. And here is the image of the micro-CT. And we can also see an obvious efficacy in the DIO data. And we can see after the siRNA drug treatment, here was a really obvious efficiency about this mice image. And here is the product list for other targets about obesity. And except for the liver target NHBE, we also have some other targets expressed in the muscles, such as GDF8, and targets expressed in the brain GDF75, and the target expressed in the adipose ALK7. So we can see the small nucleic acid drugs delivery to extra liver is also an important topic to discuss. And we will discuss this content in this part. As we know, the small nucleic acid drug have the potential to treat neurological disease, but it's challenging to deliver these drugs to other tissues, such as the brain tissue or the muscle tissues, especially the brain, because of the blood-brain barrier. And antibody oligonucleotide conjugate is an important method to treat to drug delivery to other tissues. Here is the target TFR1. TFR1 is the receptor of transferrin and functions in iron transport and through the transcytosis mechanism, and is highly expressed on the apical surface of endothelial cells in the BBB. And the process is crucial for maintaining the iron homeostasis in the brain and has been targeting for enhancing drug delivery across BBB. And except for delivery to the brain, this TFR1 has also can be used to the delivery to the muscle and to the eyes. Here is the gene targeting strategy for our humanized TFR1 mice. And in this mice, the exon 4 to 19 of mouse TFR1 that encode extracellular domain are replaced by human competitors in humanized TFR1 mice. And the genomic region of mouse TFR1 that encode the cytoplasmic portion is retained. And the promoter and the 5' UTR and the 3' UTR are also retained. And here is mRNA and protein expression data of this humanized TFR1 mice. And we can see the mRNA data by RT-PCR. And we can see here is only mouse TFR1 in wild-type mice. And here is only human TFR1 in this humanized mice. And we also conduct the protein expression analysis in our humanized mice. And we can see the human TFR1 was exclusively detectable in the humanized TFR1 mice, but not in wild-type mice. And here is the protein analysis of TFR1 in brain macrovessels by immunofluorescence. And we can see the green signal is human TFR1 and the red signal is mouse CD31, which is a marker of endothelium. And we can see colocalization about human TFR1 and mouse CD31, which means the human TFR1 was detectable on the brain macrovascular endothelium of the humanized TFR1 mice. And we also conduct a protein analysis by IHC. And we can see the red signal is the signal of TFR1 protein expression. And we can see human TFR1 can detectable in different tissues such as heart and liver and the brain, which is similar to the expression profile of mouse. Except for the expression data, and our humanized TFR1 mice have successfully been used to the drug delivery to CNS or to the muscle. And here is the paper published by our client by using our TFR1 mice. And in this paper, they discovered an AAV capsid combined with human TFR1 and mediate the brain-wide gene delivery. And in the in vivo test, they use our humanized TFR1 mice. And in this paper, they can see the AAV capsid target TFR1 can successfully deliver this AAV to the brain in our humanized TFR1 mice. Here is another published paper by our client. They also use our humanized TFR1 mice. And in this paper, they discovered an AOC drug target TFR1, and it can mediate DMD exon skipping. And they also use our humanized TFR1 mice for in vivo test. And here is the model list of TFR1 with other humanized targets for different disease areas. And here is the example of Alzheimer's disease target tau. And we have the single target humanized tau mice. And here is the gene targeting strategy. And we can see the exon 2 to exon 10 of the mouse tau that encode the full-length protein were replaced by the human tau from exon 2 to exon 15 in humanized tau mice. And the 3' UTR was replaced by the human tau 3' UTR. And in the model validation, we can see the RT-PCR results showed there was only mouse tau in the wild-type mice, and there was only human tau in the humanized mice. And in the in vivo efficiency test, these tau mice were treated with siRNA drug target tau by ICV injection. And at day 7, we collect the brain tissue to test the mRNA result. And we can see the human tau mRNA expression was significantly reduced after treatment. And we also test the protein expression by Western. And we can see after treatment, the protein expression of tau also reduced. And after we cross off our humanized tau and TFR1 mice, and we can see in the in vivo inhibitory test, after treatment, the human tau mRNA in the brain was significantly reduced in the double humanized TFR1 and tau mice. And for the muscle delivery, we also developed humanized DMPK mice. And gene targeting for DMPK is also the mouse genome region of DMPK, which encode the full-length protein, were replaced by the human DMPK from exon 1 to exon 15. And the promoter and the 5' and 3' UTR were replaced by the human compartments. And we also across our humanized DMPK mice and TFR1 mice for the muscle delivery. And in the model validation, we confirmed the human TFR1 expression in this double target mouse. And we also confirmed the protein expression of DMPK by Western blot, which use a cross-reactive antibody with mouse and human. And for the in vivo test, this double target humanized TFR1 and DMPK mice were treated with an AOC drug target DMPK. And after treated by IV injection, different tissues were collected at day 7. And we can see in the muscle tissue, the mRNA level of human DMPK was significantly reduced, which means our double target humanized TFR1 and DMPK mice could successfully be used for the muscle delivery. And here is the model list of other targets for crossing BBB. Except for TFR1, we also have the targets such as CD98HC and IGF-1R. And another part is about in vivo gene therapy. I think the in vivo gene therapy we discussed here is about for the genetic disease because of the genetic mutations or the deletion because of the genetic disease. And the method for the treatment for this gene therapy include gene addition and gene editing and base editing. So our genetic knockout mice or our humanized mutations mice can be used for the in vivo gene therapy. The first one example is for a muscle disease DMD. We know DMD is a severe progressive muscle wasting disease caused by the mutation about the gene DMD or the exon deleting. And the deletion of DMD can cause the deficiency about the dystrophin. And without dystrophin, the human without DMD are more sensitive to muscle damage and resulting in the progressive loss of muscle tissues and functions. So the treatment method of DMD include gene therapy by delivering a normal DMD by AAV, or the gene editing by CRISPR-Cas9, or the exon skipping by small nucleic acid drugs. So for the DMD research, we develop a series humanized or knockout mice for DMD research. Here is the data of our DMD knockout mice. And we can see the gene targeting strategy, the exon 45 to 50 of mouse DMD were deleted in the DMD knockout mice. And we confirmed by RT-PCR and Western blot. And we can see here was only mouse DMD in the wild-type mice, but not in the knockout mice. And in the red, here is the behavior performance in the wild-type mice and homozygous DMD knockout mice. And we can see the green strengthen results show the strengthen produced by the forelimb in the knockout mice was significantly weaker than in the wild-type mice. And the rotor tests were performed to test the motor coordination. And we can see the latency to fall was significantly decreased in the homozygous DMD knockout mice. And here is another model for DMD, which is a humanized DMD but deleted exon 45. And the targeting strategy is the exon 44, 45, and 46 of mouse DMD are replaced by exon 44 and 46 of human DMD, and without 45. And we can see the RT-PCR result shows the gene mutation we can see in the humanized but deleting exon 45. And here is the protein expression by Western blot. And in the red is the serum creatine activity result, because the creatine is a hallmark of muscle damage. And we can see in the data the creatine activity was significantly greater in the homozygous humanized DMD but deleted exon 45, compared with the wild-type mice, which means in this model, the muscle damage is significant in this model. And we also have the histopathological analysis in the wild-type mice and in this mutation mice. And we can see the histological data shows in the homozygous humanized DMD but deleted exon 45 shows the inflammation and the centrally located nuclei, which means in this model the muscle tissue damage. And another disease area is about eye disease. And the figure in the left shows the genetic and the clinical diversity of the mutation in different genes for the inherited retina disease. And one popular target is RHO, which can encode the rhodopsin, which is for the fundamental importance for phototransduction. And this slide shows the most common method for the treatment of RHO mutation, and such as the EDIT 102. And in the step 1, the endogenous RHO, no matter RHO mutation or the normal RHO will be knocked out. and they will deliver a normal RHO to replace the endogenous RHO. And based this mechanism, we develop of our humanized RHO P23H mutation mice. And in the gene targeting strategy, the promoter and 5' UTR and the mouse RHO gene that encode the full-length protein are replaced by the human competitor with P23H mutation. And in MRI results by RT-PCR and the gene sequencing, we have confirmed the gene mutation. And we also have the HE staining in white-type mice and heterozygous humanized RHO mutation mice. And we can see the different layer of retina was thinner compared with the white-type mice, which means the photoreceptors of this humanized RHO mutation mice degraded compared with the white-type mice. And although we have so many data, but we didn't show all the data on these slides. And here is my presentation today. And here I will summarize my presentation for the development of humanized models for oligonucleotides or small nucleic acid drug type or the gene therapy models. The target source is really important because it can determine which humanized mice target we develop. And our target source is come from the high market attention or the FDA approved drugs, or comes from the pipeline we focus on. And for the humanized model validation, we will analyze case by case, such as for small nucleic acid drugs. We will compare the expression level, and we will conduct the in vivo inhibitory test or the in vivo efficacy test. And for gene therapy models, we also develop the disease models in different disease areas, and we will confirm the disease phenotypes and how they can be used for different gene therapy models. And here is my presentation today. And if you have any questions.
Thank you for your time and thank you for joining this webinar by Biocytogen. That's all for the presentation by Dr. Jinyi. If you have any question to us, please drop us an email at info@bbctg.com.cn and our representative will reply you shortly. You may also scan this QR code to know more about Biocytogen from our official website. Thank you once again and I hope to see you next time. Goodbye.